our last presenter for the chapter session of the ocean is Keir Macartney. And he is from Texas, University of Texas, Rio Grande Valley. Yep. And I've walked away from my notes so you can see what he's going to talk about.
There's a lot of colors in my presentation, so I want you guys to be able to see it. Lots of pretty bar graphs, hopefully. But today I'm going to be taking you on a walk through the microbial community structure and some of the function of those microbes in Mesophotic Antipatharians found in the Gulf of Mexico. So Antipatharians, I've just got some pictures here. You can see these are some lip portals and then some sea fans over here. And that's my study subject at the moment. Okay, so you might know Antipatharians more commonly as black coral, and they're often used for jewelry because they produce a proteinaceous skeleton that kind of shines up really nicely. So they're important culturally in that sense. But they're also important ecologically as habitat engineers, particularly in habitat provision for mobile organisms. And they also form associations with various invertebrate fauna. This is particularly important in deep sea habitats because complex habitat is few and far between. But despite their ecological and cultural importance, we know relatively little about them. If you want to learn more, I really recommend Daniel Wagner's 2012 chapter in Advances in Marine Biology. Kind of became my Bible for Antipatharians while I was learning to work with these organisms. So my study area is the Gulf of Mexico, in particular the South Texas banks, which are the black dots here, zoomed in on this map. You may also know the flower garden banks, which are probably more famous than the South Texas banks, but the banks are also just as important as the flower gardens. So Antipatharians are found throughout the Gulf of Mexico in a very broad depth range, but we're going to focus on Antipatharians found in the Mesophotic zone, which is between 30 and 150 meters. And the South Texas banks are Mesophotic reefs. They're relic coral algal banks. And Antipatharians can be found in densities of up to 3 to 10 individuals per meter squared. But around these larger high relief structures that are known as the main banks, we also have low relief structures. And there are a lot of Antipatharians, gorgonians, and sponges also found in these low relief structures. And these lower relief structures, being about 1 to 2 meters off the seafloor, are actually pretty vulnerable to anthropogenic impacts. In particular, oil and gas exploration, as the first talk showed, Deepwater Horizon was not ideal for deepwater coral communities, but also in South Texas banks fisheries, there's fairly high trawling effort for shrimp. So abrasion damage can also occur in these environments to these corals. So there's an urgent need to get a baseline understanding of the health and ecology of these organisms. I am focusing primarily on the microbiome of these corals because of the coral probiotic hypothesis, which states that the microbial associates or symbionts of corals play a big role in their health and their ability to adapt to changing environmental conditions. Unfortunately, there is little information on Antipatharian microbial communities. There is one paper from the Mediterranean and then one paper from below 300 meters in the Gulf of Mexico. So nothing within the depth range that I want to focus on. So my objectives really were to characterize the microbial communities of these corals in the Gulf of Mexico at mesophotic depths. I wanted to see who was there, who was shared between the species, who might play a large role overall, but also assess regional variation, if applicable, if I had the sample size. Then I did want to understand functional attributes of the microbiome. It's great to characterize who's there, but it's really important to know what they're doing, if anything at all. Mostly I'm going to be focusing on nitrogen cycling, sulfur cycling, and secondary metabolites. In particular, I'm going to be trying to find any genes indicating antimicrobial activity. And then obviously I want to compare to other types of coral with published data. We're going to be talking about the ones highlighted in orange today because I don't have a lot of time, and some of these I haven't finished yet. So my methodology, in summer of 2016 and 2017, an ROV was used to collect 5 species of black coral on the South Texas banks or sites surrounding them. They were Antipathys atlantica, Antipathys fricata, Plumipathys panacea, Stichopathys leckeni, and not pictured Stichopathys occidentalis, but they look fairly similar to leckeni. There are some morphological differences. In 2021, I also collected some using scuba diving, but the primary focus is really looking at 2016 and 2017 to try and eliminate some of the time-based bias we might see. They're preserved in ethanol and then stored in freezers. I came along and pulled them out of the freezer and did PCR amplifying the V3, V4 hypervariable region of the 16S RNA gene, just standard metabar coding protocol for microbes. So when I got my sequencing data back, I was really excited. I slapped it all into Chime 2 and got my initial data out and plotted some very basic plots and kind of got a shock. I found that 40 to 70% of my reads in some samples were unassigned, which was not a great thing to see in your preliminary data. So after kind of fortifying myself with a beer, I went back in and looked at it in more detail, and I found that ASV1, or amplicon sequence variant, which essentially is the genetic sequence for one microbe in the context of this talk, or well, it wasn't actually a microbe in this case, actually matched best to the 12S RNA gene of Antipatharians. So I actually amplified my host DNA as well, unfortunately. So co-amplification is actually a problem for the primers that I use, which is really unfortunate because these are the primers that are used for the Earth Microbiome project. So we may need to look into alternate primers for Antipatharians. Luckily, I did sequence at a deep enough depth that I was able to recover a high enough number of reads per sample to actually continue my work. And so the first thing I like to do when I get microbiome data back is make pretty bar graphs because my simple mind can comprehend colors well. What you're looking at is a bar plot of our 5 species and seawater at the class level for the microbes. And it's a relative abundance, so 0 to 100%. What I really want to focus in on is the preponderance of the green, which is gamma proteobacteria, relative to seawater, which is here. And I guess I should introduce that this is Antipathys atlantica, because it's kind of hard to see. Antipathys furcata, Plumipathes panacea, and Stichopathys lacani at the end there, second to last, and Stichopathys occidentalis at the very last. But the important thing to draw from this is that there is a lot of gamma proteobacteria, alpha proteobacteria, and bacteroides. And those are some of the usual suspects you might see in a oral microbiome, which was great to see. At the phylum and class level of bacteria, there were significant differences between genus of coral, so between the Antipathys or the Stichopathys. But within these genuses, there were no significant differences between species. So Antipathys atlantica and furcata were not significantly different at the bacterial phylum or class level. Everything was different at the ASV level, so the individual bacteria level. What's great about this is somewhat similar to those communities found in other Antipatharian studies. Next thing I wanted to look at is diversity of these microbiomes using the Shannon's H index. What I found is relatively low mean diversity compared to seawater, but high variance in many of these samples, particularly in Antipathys atlantica and Stichopathys lacani. What I want to point out is that the topmost samples, 3 or 4 samples, were all collected at different sites and collection periods. So they aren't all coming from the same site. And they were also, several of these samples were verified by myself to be healthy when I collected them in the water. So that was interesting for me to see. I wasn't expecting such variance in biodiversity within a microbiome from samples that were sometimes collected at the same site. So the next thing I did is I wanted to produce a non-metric dimensional scaling plot to look at how these coral species plotted in space against each other. And for those of you that are not familiar with these plots, essentially each point is an individual coral microbiome. And if they're close together, they're very similar. If they're not close together, they're more dissimilar. So right here is seawater. Seawater clusters really well. Everything else doesn't really. They all overlap. And I work with sponges and other corals, and I have not found so much overlap before, which was surprising to me. So I thought, okay, maybe it's regional variation. So I plotted the same thing based on region, regardless of species. And I did find some separation, but again, there's a lot of overlap in that, and there's not a lot of clustering by region. But of course, I wanted to look at regional variation within a species. In a species, I had sample size big enough to do so, and were collected from multiple areas. I was able to do that with Antipathys atlantica on the top in graph A, and Stichopathes leckeni in the bottom graph. I did find that there were significant differences at the ASV level using a Permanova, but again, you can see that there is not a lot of clustering. Things are very different. There's a lot of variance within samples, even those samples that were collected at the same site. And then I kind of just went into bioinformatic purgatory and kept looking at it. And this is a really complex graph, but really what I want you to look at is the clustering at the top. So this is hierarchically clustered based on region, genus, and species, genus is plural, sorry, as rows, as rows. And then we have our microbes coming on as well. And what you really want to look at is, this is based on presence absence. So those blue bars on this graph essentially signal that a microbe is present. So our seawater samples in the corner here, that big kind of blue bar there, they all share lots of things with each other with those samples. But nothing else really shares a lot of different ASVs between each other. And then if you look at the top, nothing seems to cluster properly, which was surprising to me. So what we found is high variation in samples of the same species and even those from the same sites, which I wasn't expecting. So Antipatharians here don't appear to have consistent associations with 4 microbes based on this study. It's not unsurprising that local environment can affect microbial communities, but within site variance is very surprising. One explanation for this is that South Texas Bank Antipatharians exist in a highly dynamic environment. We have high currents, variable light environment due to the Nephilim layer, some temperature variation, and high concentration of particulate matter in the water column. These marine aggregates of detrital matter can settle in corals, and since heterotrophic bacteria love detrital particles, they may also settle in the coral and create these changes in biodiversity patterns we see. How much time do I have? Okay, so really quickly run through this. Nitrogen cycling between species. Nitrogen cycling can be important, but essentially what I found was that there were differences between corals, but no consistent pattern again. So that variation was also within the functional attributes of the corals. In conclusion, proteobacteria dominate. Are they important in outcompeting opportunistic bacteria? I think I need to zoom in on the bacteria that were consistently shared throughout species. There's very few ASVs. The variation appears to be driven by less abundant communities of bacteria in individuals due to this highly dynamic environment. This may assist in rapid adaptation to local environmental variables, or maybe they don't, we don't know. And I then would like to also go and collect more data. There's always more data to be collected, but also look at these microbiomes and experimental setups to look at the effective stressors. There are a lot of people that helped me with this. And sorry, I went over time.
Time for 2 questions for here.
Yeah, Beth? Yep.
Well, I had a question about the reference database that you used. Did you use an existing reference database? And do you think if you manually added sequences from bacteria that you expect to find, do you think you'd have a better taxonomic classification?
That's a good question. I use the Silva database, which is kind of the standard one to use in these studies, and it's pretty well updated. I don't know if I could actually find any more sequences that weren't already on the Silva database. Maybe if I can find my own, I would. Yep.
And like the graph you showed where you had the columns, like the purgatory one. I was wondering, like, it looks like there's a row that like is almost like colored, maybe like a quarter of the way down.
Right in here?
Up a little bit, like.
Those guys up here? So those are the gamma proteobacteria I was talking about, and particularly there are groups of the endozycomas and pseudomonas bacteria that are shared throughout almost all of these corals. Not all of them, which is great because it made me feel worried about contamination, but those are groups that I do want to look at a little bit more. Yeah.
Yeah, so through your research, have you found maybe some of the possible reasoning or the mechanisms behind this any sort of microbiome translocation or like microbiome shuffling that goes on between these organisms? Does that happen like through their life stages, or does it something that is pretty specific to individuals and they stay the way they are?
most people seem to, and I believe this, that these are fairly plastic. They can change
Okay.
over time, but some folks have looked at more in the dinoflagellate algal symbionts, so symbiodinium,
Yeah.
including corals that are an olate symbiosis. Those can change over a lifetime. The microbes are fairly plastic.
Cool.
Do you plan on doing any like experiments doing 15N or anything to stabilize isotopes to see how that relates to the genetic diversity you have?
I do have samples that I am sending out for stabilized analysis of 15N and 13C because I'd like to look at what role the microbes are playing, but also what these things are eating. So.
All right then, well.